Okay, so you can see my screen clearly.
Yes, Miss, I can see.
It shows the heading which is known as the order of protein structure. So just like we have a hierarchy in order to explain different things, for example, we say that organisms begin with the smallest structure, which is a cell, and a group of cells make a tissue, and a group of tissues make an organ, and group of organs, they function to make a system, and a group of various systems make up an organism. Similarly, since we are doing the chapter biological molecules and it's about proteins, so proteins also begin with the simplest of all the structures, moving on to slowly and gradually complicated and more complicated and more complicated. So there are 4 levels of organizational structure of proteins. And you can see those 4 levels. The simplest of all level is the primary structure, then the secondary, then the tertiary, and then the quaternary structure, right? So let's start with the primary structure. Instead of giving you the details about it, I'm going to show you this diagram. Okay. Now this diagram is actually showing you a chain of different amino acids. Remember, we had done, थोड़ा सा अगर मैं अभी नहीं recap करी, we had done this basic structure of a single amino acid. ठीक है? This was the amino acid. This is one amino acid, and then we also studied that 2 amino acids, they join together by means of a condensation reaction to form a dipeptide. और अगर बहुत सारे amino acids एक दूसरे के साथ लगे हुए हैं तो they make a polypeptide. ठीक है? So in reality, of course, when we say it's a protein, and we had actually also discussed about various functions of proteins inside the body. So we said that myosin is a protein, hemoglobin is a protein, and hormones are protein. ये हमने किया था. Collagen is a protein, enzymes are protein. ठीक है? तो whether it's any protein, कोई भी protein हो, every protein has a primary structure. And what is the primary structure of protein? It is nothing but a series of different kinds of amino acids that have been linked together by means of peptide bond. ठीक है? कैसे? I will now zoom into this diagram to show you कि ये इन्होंने एक प्रोटीन दिखाई है जिसका नाम है ribonuclease. Ribonuclease is an enzyme, and you have done this in O levels, that all enzymes are biological catalysts made up of protein. ठीक है? तो ये भी एक enzyme है और ये प्रोटीन से बना हुआ है. और when I say प्रोटीन से बना हुआ है, it means it is made up of various amino acids joined end to end together. Just follow where my cursor is going. कितने सारे amino acids हैं, right? Look at this series of amino acids, and the chain finally ends here. ठीक है? And if you look at the individual amino acids, we can see that every amino acid is unique, since you know that the uniqueness of an amino acid depends upon what? Yes, Alia?
Miss, on the way they are arranged. Miss, on the R group, Miss.
Yeah, on the R group. The uniqueness of an amino acid depends upon the R group. ठीक है? So when I say that this is lysine, what makes this lysine and what makes this glutamine, threonine, alanine, phenylalanine, arginine, histidine, methionine, aspartic acid, serine? क्यों? इसलिए कि everybody has a different R group. And just like I told you that there are 26 letters of A to Z, and from those 26 letters, we make millions of words, right? Similarly, there are only 20 available amino acids, but the way they are arranged, the way they make up a chain of a protein determines कि ये protein एक enzyme है, या ये hormone है, या ये collagen है, hemoglobin है, keratin है, जो कुछ भी है, right? अच्छा. So this is how, now you can see 2 things in this picture. And those 2 things are, number 1, you can see the names of various amino acids. Number 2, you can see that they are arranged in a very organized specific sequence. So the primary structure means the amino acids arranged in a particular order. ठीक है? अगर आपको ऐसी तस्वीर नजर आती है, आपसे कोई कहे कि can you Can you say that this picture is showing a primary structure? So your answer will be yes. Let's go to the definition now. Okay? Now you read this definition, the bullet points in 1 to 2 minutes, and then I will discuss with you. Kinds of amino acids, ek to kinds are different, and then they are arranged in a different sequence. Okay? So to sum up, I can say in very simple words, the primary structure is the sequence of the amino acids in a polypeptide. And when I say polypeptide, I mean many amino acids joined to each other by means of several peptide bonds. And a usual and average protein is usually several hundred amino acids long. And the sequence of the amino acid in a protein is determined by the DNA code. I'm not going to go into the DNA code also in detail right now, because we will be eventually doing this when we reach chapter protein synthesis, right? Let's look at some other things. Now, I will be drawing something and I will be asking you a question, okay? So, if I say that this is a protein, the only hint which I'm telling you is that this is a protein. Can you see the primary structure in this protein molecule? Yes or no?
Yes, miss, we can see the amino acids.
You can see amino acids. How can you see them? They're just circles which have been drawn.
So, miss, they represent amino acids?
They represent amino acid, but if we go back to the definition of primary structure, it means it's the sequence of amino acids in a polypeptide. But can you see in this diagram any sequence of amino acids? Can you see? I don't think so. Right? Or maybe I can also draw the diagram like this. In some books, it's drawn like this. And they say this is a protein molecule, and then they ask you a question that can you see the primary structure in this? So your answer should be no. Now look, in order to give this answer in yes, and to say that we can see a primary structure, two conditions need to be fulfilled. Condition number one, you should be able to see the amino acids, whether they are in an abbreviation, abbreviated form में हो सकते हैं, जैसे यहाँ पे पूरा नाम नहीं लिखा हुआ, but the name should be seen and the sequence should be seen. I'll show you another picture. Okay, now this is a picture and I'm going to zoom into this picture. Okay, now can you see the primary structure in this bit of the polypeptide?
Oh, yes, miss, we can.
Very good. Can you see the primary structure in this bit?
Oh, no, miss.
Can you see the primary structure in this bit?
No, miss, not in this bit.
Or in this bit?
No, miss.
No. Is there a disturbance in your room? Can you hear something?
In my room, miss?
No?
No, miss, not really.
Achha, okay, fine. Then this means this disturbance is in my, maybe the sound of traffic, right?
Okay, maybe, miss.
Achha, okay, fine. Just. Okay. So did you understand this concept of primary structure? Is this clear?
Yes, miss, it's clear.
Now we move on to the secondary structure. Secondary pe chalte hain. And secondary structure means that, see, it is very natural. जैसे बिल्कुल glucose में था ना कि although I told you that glucose can be in a straight chain form, but it is more stable for the glucose to adopt a ring form. Ring form is ज़्यादा stable रहता है, right? So same is the case with proteins. Proteins of course, protein or a polypeptide is a chain of amino acids joined end to end by several peptide bonds, but it is very natural for a protein to fold. And the reason the protein folds is because of lots of hydrogen bonds. Because if you remember, when I was doing the structure of a basic amino acid, we could see that hydrogen bonds आराम से बन सकते हैं between the electronegative oxygen and the electropositive hydrogen. हो सकता है कि वो hydrogen R groups में हो, है ना? तो हाइड्रोजन बॉन्ड्स बनने का यहाँ पे बहुत ज्यादा चांस है. एंड बिकॉज द हाइड्रोजन बॉन्ड्स आर बीइंग फॉर्म्ड, दैट्स व्हाई अ प्रोटीन मॉलिक्यूल ट्विस्ट्स. इट ट्विस्ट्स एक्चुअली. ठीक है? एंड हाउ डज इट ट्विस्ट? वी विल डू इट whenever a group of amino acids, they fold because of the presence of regular pattern of hydrogen bonds. We now say that this protein has adopted a secondary structure. Primary structure तो हर प्रोटीन का होता ही होता है, लेकिन it is very natural for every protein that has a primary structure that it will fold into a secondary structure. ठीक है? And the main reason of folding is what? It is the regular pattern of the hydrogen bond. Keyword क्या है? Regular pattern of hydrogen bond. किसके बीच में बन रहा है? Between the oxygen of the carboxylic acid group of one amino acid and the hydrogen of the amino group of another amino acid. ये बात समझ आ रही है?
Yes, miss.
ठीक है? For example, ये आपके पास amino acid 1 है, ये amino acid 2, amino acid 3, amino acid 4. तो amino acid 1 has of course a carboxylic acid group इस end पे, और इस end पे amino group होगा. ठीक है? तो और अब अगर ये सेकंड अमाइनो एसिड है तो यहाँ पे इसका NH ग्रुप होगा, फिर यहाँ पे इसका COH होगा and so on. So this description is saying that the hydrogen bonds will form between the oxygen of the carboxylic acid group, यानी इसके बीच में और इसके बीच में, फिर इसके बीच में और इसके बीच में. अब जब ये रेगुलरली होता जाएगा, so what will happen? This will cause the amino acid chain to curl on its own self, to twist. One very very important thing जो हर student भूल जाता है in describing the secondary structure is that the regular pattern of the hydrogen bonds does not involve the R groups. यानी ये जो आपकी हाइड्रोजन बॉन्डिंग हो रही है, ये R ग्रुप्स के बीच में नहीं हो रही है. It's between the oxygen of the carboxylic acid group of one amino acid and the hydrogen of the amino group of the other amino acid. So it doesn't involve the R group interactions. ठीक है? But one thing is for sure, ये regular है, ये बिल्कुल regular है. अच्छा, अब how do these proteins or amino acids fold? जब हम कोई कपड़ा तय करते हैं ना, कपड़ा लपेटते हैं. अच्छा, yes, when we fold a cloth, we can fold it in two ways. Either we can fold it into a tight fold, या जैसे हम कभी रस्सी को भी फोल्ड करते हैं ना, तो हम रस्सी को बहुत टाइटली फोल्ड कर सकते हैं और हम उसको ढीला ढीला भी फोल्ड कर सकते हैं. दिस इज अ लूज फोल्ड and these are tight folds. ठीक है? So depending upon the arrangement of these hydrogen bonds, के how many hydrogen bonds are there, क्या बहुत सारे हैं या बहुत कम हैं, तो it depends. A protein can fold into a tight coil which is known as an alpha helix, or a protein can fold into a loose sheet which is known as a beta pleated sheet. Right? Is this understandable? Yes or no? Alia?
Yes, miss.
Is this understandable?
Yes, miss, it's understand.
So I thought that you are muted. Acha, okay. So Alia, if I extend my class to 6:10, will it be a problem for you? 6 p.m. and 10 minutes more?
No, miss, it's fine with me.
Because what happens at every time on this day, which is Monday, my Maghrib namaz gets a bit delayed and I actually don't like it. So I'll be taking a break for the Maghrib namaz. Okay. अभी तक तो खैर हुई नहीं अजान, तो what you do is that you go through this information that you can see and then I will explain you. You go through this.
Okay.
Read only till here, not beyond this line. Okay?
Okay.
Are you done reading?
Miss, almost, Miss. I'm on the fourth pointer. Yes, Miss. I'm done reading.
Now, in order to explain you this further, look what they actually mean. In fact, you know, the best diagram would be a diagram from your own textbook. So let's look at these diagrams. कि इसका क्या मतलब है कि the first amino acid and four amino acids ahead of it. ठीक है, so this is actually what they mean. This is the same as the picture which I showed you. And can I ask you that, can you see the primary structure in this diagram?
Yes, Miss. We can.
Yes, you can see the primary structure, right? What two reasons will you give, Alia?
Miss, we can see the symbols, Miss.
You can see the symbols, and can you see a proper sequence?
Yes, Miss.
Yes. Okay. Now, this primary structure can fold because of many hydrogen bonds. तो अब ये क्या है कि, for example, the numbers are not given over here. Fine. Here the numbers are given. Look, I'm going to now put my pen and you look how I'm doing. Amino acid number 1, 2, 3, 4, 5, 6, 7, 8, 9. ठीक है? Every dotted or dashed pink line is denoting a hydrogen bond. Okay? So the amino acid 1 is making a hydrogen bond with 4 amino acids ahead of it. So 1 plus 4 is equal to 5. मतलब क्या? कि लेट्स से हो सकता है कि यहाँ पे होगा ऑक्सीजन ऑफ द COO group and it's making a hydrogen with the hydrogen of the amino group of 4 amino acids ahead. ठीक? अब हम देखते हैं अमाइनो एसिड जो है वो 3 किसके साथ बना रहा है. दिस इज अमाइनो एसिड 3. सो 3 प्लस 4 इज 7. सो अब देखो ये डॉटेड लाइन कहाँ जा रही है, अमाइनो एसिड 7 पे जा रही है. फिर 5 किसके साथ बना रहा है, 5 प्लस 4 9. So the amino acid 5 is making hydrogen bond with the amino acid 9. Since this is a regular pattern, है ना, ये एक पैटर्न है, ये ऐसी रैंडम नहीं बन रहे. So because of the regularity of the pattern of these hydrogen bonds between the oxygen of the CO group and the hydrogen of the amino group, such kind of a structure is known as the secondary structure and it has a specific shape. अब अल्फा हेलिक्स बनाने के लिए इसका ये रूल है जो आपको नजर आ रहा है, ठीक है, ये वाला रूल, इसलिए बड़ा टाइट सा हेलिक्स बनता है, ऐसे ऐसे ऐसे, ठीक. बट इन ऑर्डर टू मेक लूज फोल्ड्स, लूज फोल्ड्स इतने टाइट नहीं कि जितने अल्फा हेलिक्स में होते हैं. यू कैन सी नाउ हियर द हाइड्रोजन बॉन्ड अरेंजमेंट इज अ बिट डिफरेंट, ठीक है, इसमें डिफरेंट है, इसमें कम हाइड्रोजन बॉन्ड्स हैं, दैट्स व्हाई इट गिव्स यू अ लूजली फोल्डेड शीट. If you have a pattern of hydrogen bonding like this, it is known as an alpha helix. And if you have a hydrogen bonding pattern like this, it is known as a beta pleated, जैसे लगता है pleats हैं, ठीक है, pleated sheet. अच्छा, the thing that confuses almost all students of AS biology is वो ये समझते हैं कि जब वो डायग्राम देखते हैं बीटा प्लीटेड शीट का, तो वो ये समझते हैं कि दिस इज वन पॉलीपेप्टाइड चेन, दिस इज अ सेकंड पॉलीपेप्टाइड चेन एंड दिस इज अ थर्ड पॉलीपेप्टाइड चेन. ऑल्दो ये एक ही पॉलीपेप्टाइड चेन है. कैसे? ये इन्होंने आपको डायग्राम इस तरह दिखाया है, ये फोल्ड हुई है ऐसे, इट्स फोल्डिंग लाइक दिस, इट्स कनेक्टेड लाइक दिस, इट्स वन पॉलीपेप्टाइड चेन. So whether it is an alpha helix or it is a beta pleated sheet, at the end of the day, it's only one polypeptide chain. Let me go back and show you this thing. This is also a diagram of beta pleated sheet. On the left hand side, you can see alpha helix. ठीक है जी, आपको ये helix नजर आ रहा है. And then this is the beta pleated sheet. ठीक है. तो अब जो ये डायग्राम है ना, इट इज डिफरेंट फ्रॉम द डायग्राम इन योर टेक्स्ट बुक. उसमें लगता है जैसे अलग अलग पॉलीपेप्टाइड चेन्स है, अलग नहीं है, एक ही है, लेकिन वो ऐसे करके फोल्ड हुई है. एंड लुक एट द हाइड्रोजन बॉन्ड्स. ऑक्सीजन के बीच में है ऑफ द CO ग्रुप, H of the amino group, again the same, again the same, again the same, and so on. The pattern of arrangement of the hydrogen bonds in the alpha helix was between one amino acid and 4 amino acids ahead of it, whereas the pattern of arrangement of the hydrogen bonds in the beta pleated sheet is formed by the adjacent parallel or anti-parallel chains held together by hydrogen bonds. ठीक है? होगा वही between the oxygen of the CO group and the hydrogen of the NH group. What are you supposed to remember? That one polypeptide chain folded back onto itself in globular proteins. This kind of a structure of beta pleated sheets is a stable and rigid structure. It gives stability, strength and rigidity to a protein molecule. इसकी सबसे बेस्ट एग्जांपल है फिब्रोइन प्रोटीन. फिब्रोइन जो है ना, वो सिल्क प्रोटीन होता है जो सिल्क वर्म बनाता है, राइट, जिससे कि प्योर सिल्क बनता है और प्योर सिल्क के कपड़े बनते हैं, साड़ीज बनती हैं, राइट. So it's very difficult to tear apart silk, and the reason for that is that silk is made up of these beta pleated sheets, और उसको फाड़ना इसलिए मुश्किल है कि the hydrogen bonds arrangement has made it a very strong, stable and rigid structure. ठीक है? The important note that I have written in the end is that whether it is an alpha helix or it is a beta pleated sheet, the polypeptide chain is one. So don't get confused when you see the diagram. Is that clear, Alia? Yes, Alia. Alia, can you hear me?
Yes, Miss, I can hear you.
Okay. Now, Alia, I am taking a small break for namaz, Maghrib namaz, but then I'm going to give you a question. Okay, let me just stop the recording.
Miss, just one question.
Okay. Okay.
This meeting is being recorded.
Which levels of protein structure? Achha, you haven't done. First, though, you haven't done. Second one. The diagram shows two molecules of glucose. 4 possible bonding positions are labeled P, Q, R, and S, T, U, V, W. Okay, what do you think is the correct answer for this? Is that you have to actually find out okay, when these two molecules condense to form glycogen, where could the bonds form? Yes.
So Miss, for glycogen, it forms 1,4 linkage and 1,6 linkage. So I chose option B, Miss. PU and QW.
P, just a minute. Just a minute. Hmm. So you think that in glycogen there are 1,4 bonds also and 1,6 bonds also. So you chose option B. Correct. We haven't done water. Achha. Have I done food tests with you?
Yes, Miss, we did.
Achha, we did. And some of them you have done in O levels also. So in that case, what should be the correct answer?
Miss, I was getting A for this.
A. Yes, correct. When you add biuret reagent and if protein is present, तो आपके पास lilac color आएगा, मतलब कि वही purplish या violet, ठीक है? And उसके अंदर ये भी है, reducing sugar भी है, protein भी है, starch भी है, so you will get milk doesn't have starch, so milk will not give you a positive test for iodine. Milk has lactose in it, so lactose is a reducing sugar, so you will get orange precipitate. And milk has fats also, so you will get um, a positive test for fat emulsion, जिसमें कि एक milky emulsion आपको मिलेगा. तो ये समझ आ गया आपको question?
Yes, Miss.
Right? अच्छा, what about this? What's the answer to question number 5?
Miss, for 5, it's A, Miss.
Yes,
Because the OH group is facing upwards.
5 is A. The OH group is pointing upwards. Very good. What about the last question? Which statement describes how the molecular structure of starch is suited to its function?
Miss, starch is basically used for storage, so I chose option D, Miss.
Option
D.
D.
Last one.
D for dog.
Yes, Miss.
Okay. Yes, D is correct. This, did you do this question?
Not yet, Miss. I was doing this one.
अच्छा, ठीक है. तो अगर हम थोड़ा सा jump मारें और हम इस question पे आ जाएं, because we are doing this topic. So let's do it together, both of us. There are many types of amino acids, but only 20 that are polymerized to make polypeptides and proteins in animals. Name the type of chemical reaction that occurs when two amino acids form a dipeptide.
Miss, condensation reaction.
Very good, condensation reaction. Then let's move on to the next question. Figure 9.1 shows two amino acids, glycine and valine. Use the space below to make a drawing to show what happens when these two molecules join together to form a dipeptide, right? So when I want to show the formation of a dipeptide, now you tell me, I'm asking you a question. What is this?
Miss, it's the amide group.
What is this?
It's the carboxylic group.
What is this?
Miss, it's the hydrogen atom.
But the R group. Or you can call it, इसके बहुत सारे नाम हैं. You can call it the R group. You can call it the functional group. बहुत सारे नाम हैं, सब correct है. And you can call it the side chain. सब सही है. ठीक है? अच्छा. And then it also has an extra hydrogen. And this is the simplest of all the amino acids, and we call it glycine, जिसके पास R group भी hydrogen है, उसके अलावा भी जो एक constant वाला hydrogen होता है, वो तो है ही है hydrogen. Then we have this valine. Okay. What is this?
Miss, it's the amide group.
This.
Carboxylic group.
And is this the R group?
Yes, Miss, this is the R group.
If this is the R group, is this the constant feature of the amino acid, this one?
Yes, miss.
Correct. So if I want to show a peptide bond formation between these two, then I should show the peptide bond formation between the, in fact, you know, what does it say? It says use the space to make a drawing. अच्छा ठीक है, हम drawing बनाएंगे और हम carboxylic acid end भी label करेंगे और हम ये भी label करेंगे the amino end. So I can just label this as the amino group, the carboxylic acid group. Okay. Now what should I do? In order to show a condensation reaction between these two, okay, so that they give you a dipeptide, which I'm going to draw here. So is this how I draw a dipeptide? Now what do I do?
So miss, there'll be a CO double bond for it, miss.
And like
And then
this?
there'll be an NH bond.
Okay, N and H bond. So I have to show that this and this is removed. Yes?
Yes, miss.
And then when I'm showing, I should also label it. I should say Acha. Is this okay now?
Yes, miss.
So this is how a dipeptide is formed, and the mark scheme is, I'm reading the mark scheme, I'm saying that the mark scheme says peptide bond is drawn correctly. This is a peptide bond. It should be drawn correctly, and in fact you can label it also. आप इसको label भी कर सकते हैं. Peptide bond drawn correctly, amino and carboxylic acid ends shown, correct R groups drawn, elimination of water shown. And this is a question that carries 4 marks. So हमने सारे points show कर दिए हैं, so we can get full marks. Is that okay?
Yes, miss. That's okay.
ठीक है, समझ आ रही है बात? अच्छा, so I think that you know I should go. You can then finish this worksheet afterwards because I want to continue with where we were. हम इधर थे and we were doing secondary structure. ठीक है? अच्छा, अब secondary structure में जो आपकी example दी हुई है ना, वो फिब्रोइन की है. It is made up of beta pleated sheets. And alpha helix में जो example दी हुई है यहाँ पे, which example have they given? Keratin. Okay? Keratin हम करते हैं ना, keratin treatment होता है hair का, right? So keratin is actually a protein found in your hair in males, right? And if we look at the structure of keratin, तो वो बिल्कुल alpha helix की तरह है. So if you want to remember an example of a real life protein which has an alpha helix structure in the form of a secondary structure, you should quote keratin as an example. And if you want to give an example of beta pleated sheet, so you can quote silk protein. समझ आ रही है बात?
Yes, miss.
अच्छा, we've done this. बहुत सारे diagrams हैं. ये भी beta pleated sheet है. अच्छा. Okay. Can you see the primary structure?
It's in the first three.
Amino acids.
In the first. Yeah, miss.
Can you see the alpha helix? Oh, sorry. Just a minute. Let me go to that slide. Can you see the alpha helix?
Yes, miss. We can see it.
So can I say, acha, in other words, let me ask you a question like this. Can you see the secondary structure?
Yes, miss.
Can you see the secondary structure over here?
Yes, miss. Both of them are secondary structures.
What is the difference between the green colored secondary structure and the blue colored secondary structure?
Miss, the alpha-helix structure is it's helical in shape, while the beta-pleated one isn't.
Beta pleated is loose folds, right? Acha. Then we move on to the tertiary structure. हम tertiary structure भी आज ही कर लेते हैं, तो quaternary किसी और दिन करेंगे.
Has a sheet structure.
Huh? Did you say
Okay.
something? Did you say something? Yes, Alia. आपको
No,
कुछ?
miss.
अच्छा, ठीक है, ठीक है. अच्छा, अब tertiary structure कैसा है? Tertiary structure का मतलब ये है कि अगर ये सारा कुछ नहीं लर्न करना ना, इसको देख के दिमाग घूम रहा है कि इतने सारे बुलेट पॉइंट्स हम करेंगे, राइट? सो ओनली वन इजी वे टू रिमेंबर द टर्शियरी स्ट्रक्चर इज एनी प्रोटीन. जाहिर है हर प्रोटीन का एक प्राइमरी स्ट्रक्चर तो होगा, सेकेंडरी भी डेफिनेटली होगा. अब टर्शियरी इज वन लेवल अहेड. सो एनी प्रोटीन विच इज शोइंग एक्सटेंसिव फोल्डिंग एंड कॉइलिंग, थोड़ी बहुत कॉइलिंग तो उसमें पहले से ही थी प्राइमरी स्ट्रक्चर में, सॉरी सेकेंडरी स्ट्रक्चर में. But if any protein is showing extensive folding and coiling, ऐसी folding कि it folds and coils it into a three-dimensional shape, into a compact shape, into a globular shape, is known as a tertiary structure. कैसे? I'll just draw and tell you कि कैसे होता है tertiary structure. अब आप इसको देखो. ये पहले एक chain थी, you know, this was a chain which had a primary structure. It was a straight chain. ठीक है? और straight chain में we had many amino acids. ये बिल्कुल सीधी straight chain. Then this chain became twisted like this. So we say कि there are many regular patterns of hydrogen bonds, और ये हो गई आपकी secondary structure. फिर क्या हुआ कि ये chain हो गई ऐसे. Now look what's happening to the chain. What have I done to the chain? जी,
Miss, you added
more folds.
Yeah, miss.
And I'm not adding folds. In fact, let me draw a better diagram. I am going to draw it once again here. I have added folds. I have extensively coiled it and I have folded it, but I have not randomly coiled and folded it. I have coiled and folded it into a 3D structure. Into a globular shape. Into a compact shape. Or I can say into a sphere. एक sphere और circle में क्या फर्क होता है? Circle तो 2D होता है ना, sphere is 3D. So tertiary structure can be defined simply as an organizational level of protein structure in which there is extensive folding and coiling in such a manner that the protein folds onto itself to make a compact globular tertiary structure or a spherical structure. ठीक है? अच्छा, who is responsible for that? क्या क्या चीजें responsible? जाहिर है जब भी कोई प्रोटीन फोल्ड होती है तो उसमें bonds involved होते हैं. But here the bonds are different and I will be showing you all those bonds. अब ये देखो. This picture is that of a tertiary structure of a protein molecule. ठीक है? कैसे? ये जो आपकी जो है ना, ये वाली, ये आपकी पॉलीपेप्टाइड बैकबोन है. क्या मतलब हुआ पॉलीपेप्टाइड बैकबोन? इसका क्या मतलब है पॉलीपेप्टाइड बैकबोन?
Miss, the primary structure in the background.
Exactly, the primary structure in the background, and basically इसका मतलब ये है the polypeptide backbone means the constant part of the protein molecule. जो सारे constant features हैं ना, amino groups हो गए, carboxylic acid, the central carbon atom और एक जो hydrogen atom हर amino acid पे लगा होता है ना, इसका मतलब ये है. So the polypeptide backbone means the constant feature of a protein molecule. तो constant feature को नहीं draw किया, तो क्या चीज draw की है? What are they showing you? Are they showing you the R groups? है ना, R ग्रुप्स दिखाए है ना. So here they are showing
But
you.
miss, they are all different groups, right, miss?
हाँ, they are all different groups. बहुत अच्छी बात आपने नोट किया. They are all different groups. For example, let's look at these two R groups. इनको देखते हैं. एक ये रहा R group, ठीक है. अब गौर से देखो मैं क्या कर रही हूँ. एक आपका ये R group है. Another R group is this. ठीक है. और इन दोनों R groups के बीच में ये डॉटेड जो बॉन्ड बना हुआ है ये हाइड्रोजन बॉन्ड, क्योंकि this R group was electro positive and this R group is electro negative. ठीक है. तो there is a chance कि क्योंकि ये इलेक्ट्रो पॉजिटिव है और ये इलेक्ट्रो नेगेटिव है तो इनके बीच में आराम से एक हाइड्रोजन बॉन्ड बन सक रहा है. अब एक चीज याद रखो, up till now the bonds that we were talking about in secondary structure were bonds, they were hydrogen bonds, but they were not formed between the R groups. Again I repeat, जो सेकेंडरी स्ट्रक्चर के अल्फा हेलिक्स में हम बॉन्ड्स कर रहे थे वो आर ग्रुप्स के दरमियान नहीं बन रहे थे. लेकिन यहाँ पे when we are doing tertiary structure, these R groups are formed between these bonds are formed between the R groups. ठीक है. एक और आपको बॉन्ड दिखाती हूँ. अच्छा, ये बॉन्ड हो गया आपका हाइड्रोजन बॉन्ड. आयोनिक बॉन्ड, पॉलीपेप्टाइड बैकबोन हो गई आपकी. This is one R group which has an extra amino group. याद है मैंने कहा था इसके पास एक एक्स्ट्रा है तो इसका ओवरऑल चार्ज क्या हो जाएगा बताओ.
Miss, positive.
पॉजिटिव. वेरी गुड. ये वाला जो आपका अमाइनो एसिड है इसके पास एक एक्स्ट्रा कार्बोक्सिलिक एसिड ग्रुप है तो इसका ओवरऑल चार्ज क्या है.
Negative.
नेगेटिव. तो क्या इन दोनों के बीच में आयोनिक बॉन्ड नहीं बन सकता. आयोनिक बॉन्ड्स आपने किए ना केमिस्ट्री में. सो दिस मीन्स दैट इफ an R group, if the R groups are ionized or if the R groups are charged, then technically an ionic bond can form between ionized R groups. बन गए ना बॉन्ड. अच्छा, अब ये एक बन गया. एक और बॉन्ड देखते हैं हम, थर्ड काइंड ऑफ अ बॉन्ड. This is an R group and this is also an R group. Both these R groups, they contain a sulfur atom. Whenever two R groups that contain a sulfur atom lie adjacent to each other, there is a chance that there will be a bond formation between them, and that bond is known as disulfide bridge. Right? It's a disulfide bridge, meaning that it's a connection between two sulfur atoms which are lying adjacent to each other, right? और वो कहाँ से आए सल्फर एटम्स, वो आर ग्रुप के अंदर आ गए तो ये आराम से एक बॉन्ड बना लेंगे. ठीक. लेट्स स्टडी अ थर्ड काइंड ऑफ अ बॉन्ड. ये है, इसको हम कहते हैं हाइड्रोफोबिक इंटरेक्शन. कैसे? याद है मैंने कहा था कि देयर आर सम आर ग्रुप्स व्हिच आर हाइड्रोफिलिक एंड देयर आर सम आर ग्रुप्स व्हिच आर हाइड्रोफोबिक. तो जो हाइड्रोफोबिक आर ग्रुप्स होंगे ऑफ कोर्स वो नॉन पोलर होंगे, है ना. दे विल नॉट बी सॉल्युबल इन वाटर. तो अगर किसी एक अमाइनो एसिड चेन में यानी कि पॉलीपेप्टाइड चेन में बहुत सारे हाइड्रोफोबिक आर ग्रुप्स एक जगह इकट्ठे हो जाएं, यहाँ पे दो दिखाए हैं. ठीक है. व्हाट आर दोस, मैं आपको इसको सर्कल करके दिखाती हूँ. आर ग्रुप नंबर 1, आर ग्रुप नंबर 2, ये दोनों हाइड्रोफोबिक हैं. सो सिंस दे आर हाइड्रोफोबिक एंड दे आर नॉन पोलर, तो इनमें भी एक क्वालिटी होती है कि ये एक दूसरे के साथ इंटरैक्ट करते हैं. एंड सच काइंड ऑफ इंटरैक्शंस आर नोन एज हाइड्रोफोबिक इंटरैक्शन. क्या मतलब कि all these hydrophobic groups, they tend to stick to each other and they tend to push away themselves from water. तो अब एक चीज गौर से देखो आलिया. इमेजिन करो दैट दिस पॉलीपेप्टाइड चेन जो आपको बनी हुई है ना, ऐसे करके फोल्ड हुई हुई है. दिस द रीजन दैट इट वाज फोल्डेड वाज बिकॉज इसमें सिर्फ सेकेंडरी स्ट्रक्चर था. ठीक है. थोड़ी देर के लिए इमेजिन करो कि इसमें टर्शियरी स्ट्रक्चर नहीं है, इसमें सिर्फ सेकेंडरी स्ट्रक्चर था. सो द सेकेंडरी स्ट्रक्चर इज बिकॉज ऑफ द regular pattern of the hydrogen bonds, not between the R groups but between the oxygen of the carboxylic acid group of one amino acid and the hydrogen of the amino group of 4 amino acids ahead of it, जिसमें कि R group involved नहीं है. ठीक है. तो वो आपको सिर्फ और सिर्फ ऐसी फोल्ड हुई भी नजर आएगी. लेकिन अगर इस चेन में यहाँ पे एक सल्फर लगा हुआ है आर ग्रुप, फिर इधर एक सल्फर लगा हुआ है, तो इन दोनों के बीच में हो जाएगी अट्रैक्शन। यहाँ पे एक आयनाइज्ड आर ग्रुप लगा हुआ है, यहाँ पे एक आयनाइज्ड आर ग्रुप लगा हुआ है, तो इन दोनों के बीच में बॉन्ड बन जाएगा। यहाँ पे एक हाइड्रोफोबिक आर ग्रुप्स बने हुए हैं, तो ये एक दूसरे के साथ अट्रैक्ट हो जाएंगे। तो बिकॉज ऑफ द फैक्ट दैट a polypeptide chain can have so many varieties of R groups, so they will also interact with each other through the hydrogen bonds, ionic bonds, disulfide bridges and hydrophobic interaction. तो इन तमाम bonds के combined effort की वजह से the protein molecule will become extensively coiled and folded. Are you understanding this thing? If you're not, let me know.
It's understandable.
Your connection is very very weak, beta. आवाज नहीं आ रही, दोबारा से बताओ.
Miss, I understand it.
अच्छा, actually I can't hear you, beta.
Miss, can you hear me?
A little bit, a little bit. अच्छा, okay, do one thing. The slide is in front of you.
Miss, I was saying it's clear. Miss, I was saying it's
Achha, it's clear. Okay, it's good, it's clear. Now what you do is that the slide is in front of you. You read all these bullet points अच्छी तरीके से, ठीक है? And then you explain to me what you understood. पहले आप ये सारे bullet points आराम से पढ़ो. About different kinds of the bonds that are responsible for maintaining the tertiary structure. ठीक है, इसको अच्छी तरह से पढ़ो.
Miss, I'm done reading it.
अच्छा. Can you explain me now all what you have studied onto this diagram? What do you understand by the four types of the bonds and their role in maintaining the tertiary structure of proteins? Yes.
Miss, the tertiary structure is basically a 3D structure which is made by the folding and coiling of the polypeptide chains.
Okay.
And they're basically held together in shape by four bonds. So the first one is the hydrogen bonds. Yeah, miss, it is formed between the polar substances. Yeah, they're formed between the polar substances, and then the next one is the hydrophobic bond, which is formed between non-polar groups, which are basically dipoles, miss. They're insoluble, so they form the hydrophobic bond. Then there's the disulfide bond, which is formed between cysteine molecules. Yeah, so it's the SS bond. And then the fourth one is the ionic bond, which is formed between polar molecules. So it's between a positively charged R group and a negatively charged R group.
Correct. You will say that the ionic bond is formed between two ionized R groups. The hydrogen bonds is formed between two R groups which are oppositely charged. The hydrophobic interactions are due to the fact that like let's say if you have two R groups and both of them are non-polar, so they will cluster with each other, okay? And the disulfide bond will also do that. Let me show you another diagram. ये जो diagram आपको नजर आ रहा है ना, this is also showing you a tertiary structure in which these blue, this is the polypeptide chain, this is a polypeptide backbone, जिसको हम backbone कहते हैं ना, आपकी पूरी polypeptide chain जो constant feature है. ये black जो boxes हैं, these are the R groups which are pointing to the outer surface, and the blue ones are pointing to the internal surface or internal part of the protein molecules. ठीक है? होता क्या है कि आप इसको, and this is the last thing, and then after that I'm going to end my lesson. Imagine कि ये एक enzyme है, ठीक है? And you know that enzymes, they catalyze chemical reactions and they are found within the cytoplasm of the cell. So they are found in the aqueous environment. ठीक है? Enzymes के कुछ R groups जो hydrophilic हैं, जो polar हैं, ठीक है? They will face the outer part of the enzyme that is facing the aqueous environment of the cell, जहाँ पे के enzyme को participate करना है reaction में. Whereas the hydrophobic R groups, जो कि सारे non-polar हैं, insoluble हैं, they will cluster themselves in the center of the enzyme, hidden from the aqueous environment. What is going to be the benefit of this? इसका benefit ये होगा कि hydrophilic groups or the polar or the water soluble groups will point to the aqueous environment of the cell, thereby making this enzyme as a whole water soluble, whereas the hydrophobic groups will be pointing to the inside. Right? Are you getting my point?
Yes, Miss.
Right? Rest I will cover in the next class, inshallah, because I think it's enough for today. So I'm ending my class, Alia, and then we'll cover on Wednesday, inshallah, right?
Okay, Miss. Inshallah.
Okay, Allah Hafiz.
Yeah. Thank you, Miss.
You're
Allah
welcome.
Hafiz.
Allah Hafiz.